Welcome to this explainer. Biology loves a good rulebook. But what happens when the supposedly unbreakable laws of science just break? Today, we're diving into one of the greatest biological mysteries of the 20th century and looking at the brilliant minds who figured out what was actually going on when nature decided to go rogue. Let's set the scene. Gregor Mendel, the absolute titan of genetics, had given us perfectly calculated inheritance ratios. His laws of independent assortment were the gold standard. Scientists absolutely love when things fit into neat, predictable little mathematical boxes, right? Well, until they don't. As science pushed forward, those flawless mathematical rules started to show some serious cracks under the microscope.
Section 1. The genetics mystery begins. An anomaly in the ratios.
For decades, the consensus was that genes always sorted completely independently during reproduction. It just made perfect sense. But then, follow-up experiments started producing offspring that wildly deviated from the expected outcomes. The math literally wasn't adding up. Just to put this puzzle into perspective, think about the classic Mendelian ratio. When you track two distinct traits together at the same time, which is known as a dihybrid cross, you expect this neat, predictable 9:3:3:1 ratio in the offspring. That was the law. But when early 20th century geneticists actually tracked certain specific traits together, this classic ratio completely fell apart. It was a massive scientific curveball that threatened to upend our entire understanding of heredity.
Section 2. Enter the fruit fly, Thomas Hunt Morgan's perfect suspect. So to solve a mystery of this magnitude, the brilliant scientist Thomas Hunt Morgan knew he had to run his own extensive dihybrid crosses. But to do that, he needed a test subject that would give him rapid, undeniable results. Enter Drosophila melanogaster, the humble fruit fly. We have the male and the slightly larger female. They turned out to be incredibly suitable for laboratory studies, and honestly, they became the ultimate key to unlocking modern genetics.
But why fruit flies exactly? Well, they might just seem like annoying kitchen pests to us, but for Morgan, it was a highly strategic choice. Their scientific value is just monumental. First off, they're incredibly easy to breed on a simple synthetic medium right there in the lab. Plus, they reproduce exceptionally fast. We're talking a whole life cycle completed in just 2 weeks. On top of that, a single mating produces a massive number of offspring, which gives you a really robust sample size. Add in the fact that males and females are super easy to tell apart and, crucially, their hereditary variations, things like eye color or wing shape, are perfectly visible under just a low-power microscope. They were literally the ultimate genetic test subjects.
Section 3. Breaking Mendel's rules. The X chromosome discovery. Okay, so the crucial point here is what Morgan actually discovered when he put these flies to the test. He set up these crosses, combining flies with yellow bodies and white eyes against flies with brown bodies and red eyes, and then he just waited to see if Mendel's laws would hold up. Now, expectation dictated that those genes should segregate completely independently, but Morgan's actual observation revealed something completely shocking. The proportion of parental gene combinations was dramatically higher than the non-parental types. Basically, the traits were sticking together. Morgan correctly deduced that this was happening because these specific genes were physically located on the exact same chromosome, specifically the X chromosome. Section 4. Linkage and recombination explained. Redefining inheritance. To explain this massive deviation from the established laws, Morgan realized he needed a totally new vocabulary. So he coined 2 brand new biological terms that fundamentally redefined our understanding of inheritance. First up is the concept of linkage. Morgan defined linkage as the physical association of genes on a chromosome. Think of a chromosome as a long train. If 2 genes are physically sitting in the exact same train car, they're going to travel together to the next generation, right? They are linked. This completely explained why the parental traits in his fruit flies weren't separating the way Mendel's independent assortment predicted they would.
The second term is recombination. Now, while linkage explains why traits stay together, recombination describes those rare, rule-breaking instances where genes actually do shuffle to create completely new, non-parental combinations. Going back to our train analogy, recombination is kind of like the train cars briefly uncoupling, swapping some passengers with a neighboring train, and then reconnecting. It's the mechanism that generates genetic diversity.
Section 5. Cross A versus Cross B. Visualizing the invisible.
Now, what's really fascinating is how Morgan realized the story didn't just end with identifying that linkage exists. Looking at his data from these dihybrid crosses, he grouped his findings into 2 distinct categories.
Cross A tracked the yellow body and white eye traits, while Cross B tracked white eyes and miniature wings. Both sets of genes are on the X chromosome, but they behaved incredibly differently from one another. Morgan looked at the results and deduced that the genes in Cross A were tightly linked together. I mean, they almost never separated. However, the genes in Cross B, while still sitting on the same chromosome, were only loosely linked. They separated and formed new, non-parental combinations way more frequently. But how did he know this for sure?
The smoking gun is absolutely in the math. For the tightly linked genes in Cross A, again, the yellow body and white eyes, Morgan observed a microscopic recombination frequency of just 1.3%. Think about that. That means 98.7% of the time, those traits stuck together exactly as they were in the parents. They were virtually inseparable.
Now contrast that with Cross B, where he tracked the white eyes and miniature wings. These loosely linked genes resulted in a whopping 37.2% recombination rate. They were on the same chromosome, but they were acting almost as if they were independent. Because they were so loosely linked, there was just far more physical space between them for our metaphorical train cars to decouple. This huge difference in the numbers was a massive clue about the actual physical architecture of DNA. Section 6. Mapping the Genome, Alfred Sturtevant's Conceptual Leap. Let's see how this all builds into an incredible legacy, and we owe this to Morgan's brilliant student, Alfred Sturtevant. Sturtevant looked at his professor's recombination percentages and saw something totally revolutionary. He made a genius conceptual leap. He realized, wait a minute, I can use these varied recombination percentages as a literal measuring stick. If genes recombine only 1.3% of the time, they must be physically sitting right next to each other on the chromosome. But if they recombine 37.2% of the time, they must be sitting pretty far apart. By systematically measuring these frequencies, he realized he could map out exactly where genes sit on a chromosome. He created the very first genetic map.
It is absolutely astounding to think that the foundational genetic maps of today, the maps that literally allow us to understand human disease, engineer crops, and trace evolutionary history, began with a single mathematical anomaly in a tiny fruit fly. The supposedly unbreakable laws of genetics didn't actually break, they just evolved in complexity. It's just amazing what we can learn from this explainer's source material. As we wrap up today, I want to leave you with this. With modern genomics advancing at lightning speed, how far will genetic mapping take humanity next? Keep questioning the anomalies.